Hey Clayton, what's up?
Hey, I love your hair.
Thanks! It's in my jeans.
You're not wearing any jeans. Hey, Allie, do you want to come to my place to swim later?
Clayton, you don't have a pool.
Yes I do. Jean pool. Hey, Allie, how's it going?
Hey Clayton, where are your jeans?
In my DNA.
Get out. Leave. Hi, I'm Lydia MAINZER and welcome to this episode of 300 Seconds of Science. Today we are talking about deoxyribonucleic acid, or DNA. So, what exactly is DNA? DNA is the hereditary material in humans and other organisms. You have DNA, everyone has DNA. It contains your genetic instructions. DNA is used for growth, development, functioning, and reproduction. You may be wondering how DNA came to be. We are traveling back 148 years to when DNA was first isolated. 1869 was a life-changing year for genetic research. Swiss chemist Friedrich Miescher first saw DNA material inside human white blood cells. Following Miescher, Russian biochemist Phoebus Levin made great progress. Levin was able to make incredible leeway on DNA research. He discovered the order of three major components inside the nucleotides of DNA: phosphate, sugar, and a base. He was the first to identify how RNA and DNA molecules are put together. Although Levin wasn't quite sure in the terms of structure, scientists still learned from it. In fact, we learned nucleotides have different structures and categories. DNA is a code represented by letters. Instead of 26 letters, DNA only has 4. Altogether, it makes a book of your genetic information. Human DNA is made up of 3 billion base pairs, and 99% of those base pairs are the same for everyone. Another person along the discovery path was Austrian biochemist Erwin Chargaff. He wanted to learn more about the chemistry of nucleic acids, especially how DNA differs in different species. It was confirmed that there were relationships between these base pairs. Adenine always pairs with guanine, and cytosine always pairs with thymine. But we didn't know why this relationship existed. Chargaff's research on DNA base pairs helped two future scientists in DNA research. At the University of Cambridge, James Watson and Francis Crick identified its molecular structure as a double helix. Most DNA molecules contain 2 strands that are coiled around each other to form a double helix. DNA is like a twisted ladder. Sugar and phosphates form the sides of the ladder, while the variety of base pairs form the rungs. All the pieces in DNA research were finally coming together. Since 1953, the science of DNA has evolved. However, some things remain true. DNA is a double-stranded helix. The 2 strands are connected by hydrogen bonds. DNA base pairs are connected by hydrogen bonding. DNA plays a crucial part in our lives, especially when it comes to life and health. The sequence of bases in DNA helps determine the information that builds and maintains an organism. DNA tells us many things, like hair color, eye color, and diseases. DNA is also used to help solve crimes. In 1990, scientists started the Human Genome Project, a project that would map all of the genes in human DNA. The goal was to be able to read the genetic blueprint of building a human being. A genome is an organism's complete set of DNA. The human genome contains around 3 billion base pairs. They reside in the 23 pair of chromosomes that reside in the nucleus of all our cells. Sequencing is the process of determining the exact order of base pairs. The Human Genome Project was completed in April 2003 with the help of volunteers. Looking towards the future of the Human Genome Project, researchers and scientists have many objectives. They would all like to understand how the manual of the human body works, especially how all the parts work together. The mission is to achieve 3 main goals. 1. Help medical science develop effective diagnostic tools. 2. Better understand the health needs of people based on their certain genetic makeups. Lastly, to help develop new technologies to help cure diseases. Stay tuned to find out the future advancements in science. Be sure to like us on Facebook, follow us on Twitter, and subscribe to our YouTube channel at ZTV 300 Seconds of Science. I'm Lydia MAINZER, and that was 300 Seconds of Science. So go out, enjoy life, and stay curious.
This program was produced by ZTV at the University of Akron. To find out how you can make Emmy-winning media, visit the UA School of Communication online. ZTV. Make media. Make a difference.